Now, if we talk about diseases, there are two classes of diseases. One is a communicable disease, another is a non-communicable disease. Now, communicable disease is an infectious diseases, and these are the diseases that can be transmitted from infected to a healthy person. For example, COVID-19. And they're mostly caused by the pathogens like bacteria, viruses, and fungi. Examples are AIDS, flu, and cholera, and communicable diseases generally spread and develop very quickly and are acute. On the other hand, non-communicables are the non-infectious diseases. These diseases cannot be transmitted from infected to a healthy person, and they are mostly caused either by an abnormality in a cell, tissue, or organ, or due to deficiency of nutrition. Example, diabetes and cancer. These non-communicable diseases develop over a long time, but they last longer and they are chronic. Okay, so you should know the differences, examples, and the causes of communicable and non-communicable diseases. Now, this is a very general concept, like what are the factors affecting health? The first is the diet. If the person is not taking proper diet, he's prone to obesity, cancer, diabetes, malnutrition, and the deficiency diseases like scurvy, night blindness. They are linked to diet. Stress, mental stress can cause diseases like cancer, diabetes, and heart problems. Living conditions like if there's not access to proper health care, clean food and water, age, gender, all these affect health. Now, all these factors are interconnected. And the disease can be due to an interaction of these factors. So maybe due to a work stress, a person is not taking a healthy diet, that is predisposed to many health complications. Now, as we said, that communicable disease are caused by pathogens. So you should know what are pathogens. So pathogens are the microorganisms that causes a communicable disease. Example, bacteria, virus, protist, fungi. And pathogen can be transferred from a infected to a healthy person through air, water, and food. Now you should know what are the differences between bacteria and virus. So bacteria are microscopic, unicellular, prokaryotic, found everywhere from air, water, sea, human intestine. We have some good bacteria which help in digestion and making us curd. They help in decomposition. They're good for the environment. But there's some bacteria which release toxin and cause diseases. Virus, on the other hand, are exception, as these are non-living as soon as they are outside the living organism. Once they enter the living organism, they become living. They take over the host machinery, use the cell resources to make copy of themselves, and damage and destroy the cells. Now, how does a pathogen spread? There could be the lifestyle and the living conditions that can cause the spread of the disease, like cholera, diarrhea, food poisoning. They are caused by contaminated food and water. Some pathogens, they are spread by sexually transmitted, like AIDS, gonorrhea. And there are some diseases like coronavirus, flu, tuberculosis, cold. They are spread through air and droplets. So these are the various ways how the pathogens are spread. And they can ask you these questions like, what are the ways in which the pathogens are spread from the infected to a healthy person? Now we'll be looking over each of these pathogenic diseases one by one. We'll be looking over a viral disease in plants and animals, a bacterial disease, a fungi disease, the Protist disease. Now, what are viruses? The virus, as we said, are pathogens and cause infection and diseases. They are at the borderline of living and non-living. They do not have a regular cellular structure. If you just see the structure of the virus, it looks like something like this: a protein code with a genetic material, DNA or RNA inside it. They have genetic material enclosed in a protein coat. They take over the whole cell machinery of the host and make more copies of themselves and damage the host cell. Now, no antibiotics works on them. And the only management for viral disease, as you can see in case of corona, is isolation, vaccination, and some antivirus or antiretroviral drugs. Now, you should know some examples of the animal viral diseases, which is measles and AIDS. For the plant viral disease, we have tobacco mosaic virus. Now, measles is the disease caused in animals, or HIV and AIDS. Measles is an infectious disease. It causes skin rash and high fever. It is an airborne and caused by inhalation of the droplets with the pathogens. No cure is available. The diseased person is isolated, and there's a vaccination that provides an immunity. HIV is a human immunovirus which causes acquired immunodeficiency syndrome. This disease targets the person's immune cell and destroys the immune cell, so a person with AIDS can even die with a simple flu disease. This disease, which is AIDS, is spread by sexual contact, blood transfusion, or sharing needles, and these diseases can be prevented by using condoms, contraceptions, or fresh needles, and the disease is managed by taking an antiretroviral drugs. In plants, the disease is the tobacco mosaic virus. It's spread by the contact between the diseased crop with a healthy crop, or it can be transmitted by insects or vectors. The virus damages the cell, causing discoloration of the leaves, reducing the yield and growth of the crop. There's no cure and treatment available. Good pest control and good agriculture practice or growing the disease-resistant crop helps in the management of the disease. So you should know the symptoms, cause, and what happens in the viral disease in plants and animals with these examples. Now let's come to the bacterial disease. Now, if you look at the structure of bacteria in the cell biology, we have discussed the structure. You should know the structure of the bacteria and all these labelings. These are microscopic, these are unicellular, they are prokaryotes, they are found everywhere from air, water, sea, and human intestine. The good bacteria help in digestion, making curd and decomposition. The bad bacteria multiply rapidly and release toxins which cause diseases. Antibiotics and vaccination helps to treat bacterial disease, but antibiotic resistance is a bigger challenge for treating bacterial disease, and we'll see this later. Now, bacterial disease in animals, first is the food poisoning and the gonorrhea. The food poisoning is caused by salmonella. It is transferred from uncooked meat or eggs and disturbs the natural bacterial population in human intestine, causing food poisonous. The symptoms of food poisoning include vomiting, stomach ache, abdominal cramps, diarrhea, and fever. The management of the disease include vaccinating the chicken for salmonella, properly cooking chicken or meat, and separating the raw chicken separately. Gonorrhea is a sexually transmitted disease. It is caused by unprotected sex. The symptoms include discharge from vagina, the penis, pelvic pain. Long-term effects include ectopic pregnancies and infertility. It is treated with antibiotics. Contraception and limiting the number of sexual partners prevents the spread of the disease. For the plant, the disease you need to remember is the crown gall disease, which happens in Agrobacterium tumefaciens. The bacteria inserts the plasmid into the plant cell and create a mass. Can you see this? This is a genetically modified mass of undifferentiating cells known as gall, which develops at the junction of root and shoot. The bacteria is used in genetic engineering to create genetically modified cells, as scientists use them to insert the genes of their interest into the host. Okay, now let's come to the fungal disease. In animals, the fungal disease is an athlete's foot disease. In plants, it is a rose black spot. So you can see these are the black spots which are created in the leaf. So the fungal disease, the damaged heart valves can develop fungal disease. Some fungal disease in human can affect the brains and the lungs, and they are treated by the antifungal disease. Now, what is a rose black spot disease? This disease is spread by wind carrying the spore of the fungus. Once the spore land on the plant, they're spread to other parts by wind and the rain. These diseases cause purple black spots in the rose leaves, discoloration of the leaves. The leaves are unable to photosynthesize and the plants do not grow and flower. The disease is treated by spraying with fungicides. In winters, the spore stays dormant in the stem, so farmer can remove them to prevent the spread of the disease. Growing fungal resistant crop and the fungicides are the only way to prevent the disease from spreading. The Protist disease, protists are single eukaryotic, and you just have malaria in your syllabus. They require a vector called mosquito. Now, symptoms of malaria include fever, shaking. The disease is cured by anti-malarial drugs and preventing the growth of mosquitoes in using the insecticides. Now, malaria is caused by Plasmodium, but the vector is female Anopheles mosquito. Now, the protists reproduce sexually in mosquito and asexually in humans. So, the mosquito first injects the protist into a healthy person, which goes into the liver of the healthy person and produce many cells, which goes into the red blood cells and make gametocytes. The mosquito then consumes the blood with gametocytes, and the gametocytes then cause sexual reproduction in the mosquito, making more copies of the protosporozoites, and the cycle repeats. So this disease is spread using the mosquito because the part of the malarial cycle is completed in the mosquito and part of the human. So if you control the growth of mosquitoes, it can help to prevent this disease. Now, this is a little practical questions on how to culture the bacteria. Now, to culture the bacteria, we have 3 steps: sterilize, inoculate, and incubate. Now, what are the equipments we require to culture the bacteria? We require a burner so that we can sterilize everything we use by heat to kill any other bacteria or pathogen which is there. This is an inoculating tube. It's a long rod with a wire which is rounded in the end that is used to touch the culture, and then you can streak it, dot it wherever you want to grow the bacteria. The bacteria are grown in these glass plates which are petri dishes. They have a medium called the agar medium. Agar medium is a culture medium that contains carbohydrates for energy, nitrogen for proteins, and other minerals which helps the bacteria to grow. Now, first we sterilize the petri dish, agar medium, and the loop on the Bunsen burner to kill any contamination. That is the first step, which is sterilize. After sterilization, the loop is dipped into the bacterial culture and the colony is streaked on the plate. So you can see we are doing some streaking on the plate. The petri dish are then taped to prevent the contamination for incubation. They are not sealed, as sealing will stop the oxygen. Now we put the dishes on upside down to prevent any condensation, and we incubate it at 25 degrees. Now this is not the ideal temperature for the bacteria to grow, but if you use greater than 25, the bacteria will grow so rapidly, and even some harmful bacteria or pathogen can also grow. So this is a very important exam question. We incubate it at 25 degrees to prevent the overgrowth of the bacteria so that they can be easily managed and to prevent the growth of the pathogens. Now, there is some bacterial culture calculations in your syllabus. You should know that after every division, the bacteria doubles, right? So if you want to calculate the number of bacteria after each division, you need to first find out how many divisions are there, and 2 to the power division is the number of bacteria. So for example, if the bacteria divides every 20 minutes, calculate the number of bacteria after 5 hours. Now first, let's see in 5 hours, how many minutes are there? 300 minutes. So the bacteria is dividing every 20 minutes. It means that in 300 minutes, it will do 15 divisions. And the formula for the number of bacteria after each division is 2 to the power n, where n is the number of division. So if we do 2 to the power 15, we'll get 32768 bacteria in 5 hours. That's a big number. Okay, now how do we prevent bacterial growth? This is very important because we know there are a lot of diseases which are caused by bacteria. So it's very important for us to control the growth of bacteria. First, we use the antibiotic. Antibiotic can be natural or the synthetic drug. It is used to kill or prevent the growth of bacteria in a living host. Example, penicillin. Antiseptics like Savalon, betadine. It is a chemical which kills or inhibit the growth of microbes when you are applying them on the living tissues or the skin. Example, Savalon. Disinfectant is used to kill or inhibit or prevent the growth of bacteria on the non-living surface. So we sanitize all the working tables, all the non-living things. So we use the disinfectant. Antiseptic and disinfectants can only be used externally. To kill the infection internally, we have to use the antibiotics. Now, how antibiotics work? For example, penicillin. Antibiotics kill the bacteria without harming the host cell. Why? Because antibiotics, they target the bacterial cell wall and kills the bacteria, making the person feeling better. Painkillers and analgesics, they're used to create the symptoms like pain and fever, but does not kill the disease. To kill the bacterial disease, you have to use an antibiotic. Okay, now we discussed at the start that there's a problem with the antibiotic called the antibiotic resistance. Now, what happens in the antibiotic resistance? This is usually common when people do not complete the course of antibiotics. Doctors recommend them to do at least a 3 or a 5 day course for an antibiotics, but after taking 1 or 2 doses, they feel better and they stop taking the medicine. Now, what happens when they stop taking the medicine? If you look at this figure, there are 1 or 2 bacteria which become resistant. Now, what will happen? All the non-resistant ones will die, but these resistant ones will not die because they're resistant to the antibiotic. Now, when they are living, they get a chance to multiply themselves. And over a period of time, they'll multiply and make huge copies. Now, the entire bacterial population is now resistant, and that antibiotic cannot work. So antibiotic resistance is a big problem. So people are stopping the use of antibiotics in the UK, and they are only given when they are very, very essential. Now, you have to remember this, antibiotics do not work against the virus. Now, this is a question where they can give you that we have used 3 antibiotics. You can see A, B, C, and D, which is more effective. Now, you can see that D has the highest clear zone where no bacteria is living. So it means D is the most effective antibiotic. Now, how do you work out the area of the effectiveness of these antibiotics? You measure the diameter of the disc, and you can use to work out the radius and use pi r square, or you can just do pi d square over 4. Okay, so you can look at the figure. It's an exam question. They can ask you to look at the figure and tell which is the weakest antibiotic, which is the most effective antibiotic. So you can look at the area, and they can ask you to do some mathematical calculation and find the area of effectiveness, where you have to use the area of the circle, which is pi r square or pi d square over 4. Okay, now let's do a little bit on history of microbiology. There was a scientist called Ignaz Semmelweis. He was a doctor, and he found that diseases are caused by some kind of infectious agent. He found that women died during the time with the fever after giving birth because the medical students were not washing hands before assisting in delivery, and when they started washing hands, the death rate of the women would decrease. So he found that there's an infectious agent that is getting transmitted. Then there was a Louis Pasteur. He showed that infectious agents are microorganisms, and he developed vaccines for anthrax. And there was Joseph Lister. He started using antiseptic in the operations theater to kill the microorganism. Now, how you can prevent the infections? You know the causes of infection, the contacts. So you can quarantine or isolate, prevent the growth and spread of vectors like mosquitoes, vaccination, sanitization and disinfection, cooking the meat thoroughly and keeping raw and uncooked food meat separately, and promote hygiene and sanitation. Okay, now we have looked out what are the different diseases, how do they affect the body, now how your body fights against the diseases. We are not taking medicines every time, and that's also not true that we are not having bacteria or pathogens entering our body. They keep on entering our body, and our body defense mechanism or immune system is fighting against it every day. Now, how does that work? It works because the human has their own defense system, and we can classify the defense responses in humans in 2 categories: the non-specific immune response and the specific immune response. Non-specific immune responses, no matter whether the disease is just a small dust particle or a big deadly virus, the first line of defense works the same way. So these are the skin, secretions in the skin, the mucus in the respiratory tract, the hair and the mucus in the nose, the stomach acids. Then the second line of defense are your phagocytosis, which is the white blood cells, which comes and eat away the infections. Now, after these 2, first and the second line of defense of the non-specific immune response, the infection does not go away. Then your body comes with a third line of defense, which works with antibodies, and that is the adaptive immunity or the specific immune response. Now, let us look at each of these lines of defenses in detail. Now, the first line of defense is your skin. Skin is the physical barrier It is waterproof. It prevents the entry of microorganisms. The skin produces sweat and other antibacterial secretions. Any cut or wound lead the platelets to clot the blood quickly to prevent the entry of microorganisms. There are hair and mucus in the respiratory tract that trap any infections coming with breathing and prevent the microbes from entering the bacteria. There are cilia, which are hair-like structure which lines the trachea and the bronchus, which are tiny hair-like structure which beat the mucus to the throat where it is swallowed and it reaches the stomach and is digested by the stomach acid. And you also know that stomach produces hydrochloric acid. So if any infection enters the food, it is killed with the hydrochloric acid produced by the stomach. Okay, so I hope this first line of defense is clear to you. Now, after the first line of defense, we have the second line of defense, which is the phagocytosis. Now, what are phagocytosis? We have the white blood cells, which are the soldiers of the body. They prevent us against the infections. Now, if the infection still exists and it is not killed by the first line of defense, then the white blood cells do the following to kill it. First, the phagocytosis, then produces antibodies and the antitoxin. Now, what happens? These phagocytes, they are attracted by the chemicals which are secreted by the pathogens. They engulf the pathogen into a vacuole, and that vacuole is known as a phagosome. Inside the phagosome, the lysosomes comes in and it releases a lot of hydrolytic and the digestive enzymes which digest the pathogen, and then the digested debris is thrown out of the cell. This mechanism is known as phagocytosis. Now, our immune system is very active, but at times we need to trigger it. We need to stimulate it to be active. For that, we have vaccination. Now, what is vaccination? Vaccination is a dead or a weakened form of a disease which stimulates the body to make the antibodies against that disease without making us seriously ill. So we expose the person's body with the dead and the weakened pathogen. The body thinks it to be a disease and start producing antibodies. Now, the best thing that our body do is it has the memory cells of the white blood cells. They remember what action they took for that particular pathogen. So if the person is exposed to the actual disease anytime during the lifetime, the memory cells will remember that and immediately take action against them and produce a heightened response, high concentration of antibodies, and quickly wipe the disease. Even at times you don't even feel the symptoms. So we have the vaccines available for tetanus, diphtheria, for viral disease. We have measles and mumps, and smallpox is completely wiped with the use of vaccine. Okay, so vaccination is very, very important. All right. Okay, now let's move on to the next slide. What is herd immunity? Herd immunity is that large population need to be immunized to prevent the spread of the disease. If only few people are immunized, it will not help because the disease can still spread. Okay, now what is drug discovery and development? We need drugs to cure us from the disease. Like penicillin is a drug obtained from the microorganism Penicillium. Fleming observed that in his bacterial culture plate, as the penicillium was growing, the bacteria were not growing. So he extracted penicillin from the mold, and it is used to cure bacterial disease during World War II. Microorganisms and plants are the major sources of the drug. Now, what is the phases of the drug testing? Why we need to test the drugs? First, we devise the drug and we test it, whether it is effective, safe, stable, effective against the target. Okay. Now, how do we do the drug testing? We test them first on the cells or tissues and the computer model. If they pass that, then we test them on animal, which is called the preclinical test. And after the animals, we test them on humans, and that is known as clinical testing. Now, how do we do the clinical trials? We use two techniques called the placebo and the double-blind trials. Now, what is placebo? Placebo is giving an inactive version of the drug where the patient thinks that they are getting the drug, and that helps to eliminate any psychological effect that a person is getting in the treatment. And in the double-blind trials, the doctor and the patient doesn't know whether they are getting an actual drug or the placebo. It is a scientist who allocate the people into group and has the information what each group is getting, so that the chances of manual errors can be avoided. Monitoring and publishing of the result is very, very important. Now, the drug that we get from the plants, you need to remember these examples. The digitalis and digitoxin is the drug extracted from foxglove plant. It is used to create heart problems. The bark of willow tree is a source of aspirin, which has anti-inflammatory and pain-relieving properties. Okay, now you need to look over some of the plant diseases. The plant diseases like aphids. Aphids suck the phloem sap and take all the food of the plant, damaging the plant. They are also vectors of many infectious disease and transmit bacteria and virus from infected to a healthy plant. So to control the aphids, we have to use pesticides or biological pesticides like ladybirds. Nematode lives in the soil. They eat the plant roots and damage the roots of the plant. Other diseases in plants are nitrogen deficiency. Plant take nitrogen as nitrates. They need nitrogen for protein synthesis. So deficiency will lead to stunted growth and low crop yield. Magnesium is required for chlorophyll synthesis. So if there's a deficiency of magnesium, it will lead to chlorosis, in which the leaves turns yellow and the plant yield decreases due to the lack of photosynthesis. All the mineral deficiency are treated by giving the fertilizers. Now, what are the plant defense responses? We did the defense responses in the animal, the first, second, the third line of defense. In plant, the first defense, we have the cell wall. It protects the plant and restrict the entry of pathogen. Then the leaves have the waxy cuticles, which also restrict the entry of pathogen. The leaf bark is dead cells of the bark that restricts the entry of the pathogen. The leaf fall, the deciduous tree, shed the leaf that takes off any pathogens in the plant, and certain plants like mint and witch hazel, they release chemicals like antimicrobials that prevents the entries of the pathogen. Now, the other defense response against the herbivores is the foxglove plant, which releases a poison that prevents the herbivores attack. We have the plant like cactus, which has thorns to prevent the herbivores attack. Then the plants like lamp ears have hairy stems and leaves that prevents the herbivores attack. Some plants also release poisons with the hair, which prevent the attack of other animals. Some other plant defense responses is they droop or curl, like the touch-me-not plant. They droop or curl with a touch, frightening other animals. And some plants show mimicry. They mimic to be unhealthy, or they have spots of butterflies on them, so they prevent the insect attack. Okay, so I hope the plants and the animal defense responses are clear to you. Now let's come to the last part. What are monoclonal antibodies? This is a very important exam question. You should know what are monoclonal antibodies and what are their advantages. So monoclonal antibodies are the antibodies produced from a single clone of cell. Now, antibodies are produced by the white blood cells, and these are the lymphocytes that produce the antibodies. But if we have just one cell that produces all the antibodies, that will be monoclonal. Now, how do we produce these antibodies? We take the mouse and give the antigen to the mouse. The mouse blood will have now the antibodies against that antigen. So we isolate the spleen cells and remove a B cell from the mouse blood. Then we fuse this B cell with the tumor cell. Now, what is a tumor cell? A tumor cell has an ability to divide continuously. So as my B cell will fuse with the tumor cell, it will make a new cell called the hybridoma. And these hybridoma, as they are fused with the tumor cell, they have the ability to divide rapidly, and they'll make clones of my single B cell, and that will make my monoclonal antibodies. Now, where do we use these monoclonal antibodies? They're used in pregnancy testing. Now, what happens in the pregnancy testing kit? The urine of a pregnant woman contains a hormone called HCG, which is human chorionic gonadotropin. The pregnancy kit contains the monoclonal antibodies that binds to the HCG hormone. So if the urine has HCG hormone, it will bind to the monoclonal antibodies and make a color change, and that will give a positive test. The monoclonal antibodies binds to the antigens of the pathogen. So if the patient blood is added to the antibodies of the disease and it results in binding, that can help us to diagnose that a person is suffering from that particular disease or not. Monoclonal antibodies can also be used to monitor the concentration of a drug or a hormone. They can also be used to locate a chemical in a cell. The monoclonal antibodies, they are very specific, and so they can target the specific diseased cancer cell without affecting the healthy cell. They are specific, so they can cure many diseases, but they are expensive, need expertise, and they have side effects as they're produced by mouse, so they have side effects in humans. Now, this is also a very important topic, how monoclonal antibodies can help in cancer treatment. Now, monoclonal antibodies can bind to the antigens on the cancer cell, make them more exposed and trigger the immune response so that the white blood cell can kill them. So the first thing they can do is exposing the cancer cell. Second thing is they can bind to the cancer cell and block the receptors of the growth hormone. So the growth hormone can no longer bind and stop the division of cancer cell, and the cancer cell can stop there. So they can block the receptors. The second thing they can do is, as they're highly specific, they can bind to the cancer cell and release the drug to kill the cell. So since the monoclonal antibodies are specific to an antigen, they will only bind to a cancer cell without harming the healthy cells. Okay, so I hope this topic is clear to you. There are a lot of key terms and definition in this topic. So make sure you can define what is a disease, communicable disease, non-communicable disease, pathogen, culture medium, agar gel, mutation, antiseptic, disinfectant, vaccine, chlorosis, antibiotics, clinical trials, placebo, double-blind trial, monoclonal antibodies, hyperdoma. You can pause this video, try to define these terms, and come back and check the answer. So disease, as we said, is a disorder or an abnormality which produces sign and symptom, and health is a state of physical and a mental well-being. Communicable disease is an infectious disease transmitted from infected to a healthy person. Non-communicable disease are not transmitted. Antiseptic is a chemical which inhibits the growth of microbes. Disinfectants use and kill to prevent of growth of bacteria on non-living surfaces. Antibiotics are the drug which inhibit the growth of bacteria inside the living host. Pathogens are the microorganisms that cause a disease. Culture medium is a growth medium where microorganisms grow. Agar gel is a jelly-like carbohydrate which is used to grow the bacteria. And mutation is a change in the gene or the DNA. Vaccine is a dead or weakened form of disease which stimulate the body to make antibodies without making us ill. Chlorosis is the loss of green color in the leaf, which is caused by lack of chlorophyll due to mineral deficiency. Clinical trial is the testing of drugs on the group of people to validate its effectiveness. Hybridoma are the hybrid cell produced by fusing B cells with the tumor cell. Placebo are the substances which mimic the actual drug, but they are not the drug. Double-blind trials are the clinical trials in which neither the patient or the doctor know who is getting the placebo and who is getting the drug. Monoclonal antibodies are the antibodies made from clone of a single B cell. Okay, so I hope this topic is clear to you. As always, the next step would be to check the specification to make sure everything is crystal clear to you and do the exam questions on this topic. We have the link in the description box below. Please do not forget to subscribe to my channel, like, comment on this video, and do click on the bell icon so that you get notified as soon as I put a new video. You can follow me on Instagram at Expert Guidance, where I put daily dose of revision tips and important key terms every day. And do join my Facebook group, which is Get a Grade 9 in GCSEs, which is a free group where I also share a lot of resources for maths and science every day. And I have also devised a 15 weeks free GCSE revision course, which is absolutely free, where you can get all the resources for preparation of GCSE maths and science topics in 15 weeks. The link is mentioned in the description box below. If you are interested, you can join that. It's absolutely free. So I'll see you next in my next video. Till then, happy revising.